Hello all, welcome to this video lecture on pulsed electric field. Let's get started. The second methodology of interest under new preservation technologies is pulsed electric field. We're making use of short pulses of electric field when the food is kept in between two electrodes to minimize the microbial load and extend the shelf life of the product. When you place food containing microbial cells between two electrodes, you apply an electric field, then what you see is that the transmembrane potential of these microbial cells they change. Now transmembrane potential is nothing but the amount of energy or the work done in order to move a small amount of proton across the cell membrane. We have seen that cells they are made out of various ions. You have various ligand ion channels and you have ion channels which are very important for carrying out metabolic functions as well as for maintaining the equilibrium within cell. Now, when you apply electric field, this equilibrium is disturbed. The transmembrane potential is disturbed, which means that the ion channels are disturbed. As a result, what happens is that when the value of the transmembrane potential is greater than 1 volt, there is pore formation in the microbial cell, ultimately resulting in cell death. When we talk about electric field, the impact of electric field on microbial cell is function of field intensity, treatment duration, number of pulses that we are going to apply, the pH of the medium and temperature. All these factors, they are always interrelated and contribute to the microbial cell death. Usually for vegetative cells, we make use of an energy of around 15 kilovolt per centimeter. But if we want to target spores, let's say we can increase it to up to 80 kilovolt per centimeter. Now, very important concept to remember with regard to this is critical field power. Critical field power is that value of electric field at which the transmembrane potential of the cell, microbial cell, is greater than 1 volt and it is capable of initiating pore formation in these cells. Critical field power, it depends on the form of cell that we are dealing with, whether it is gram positive, gram negative, yeast, bacteria, fungi. Then it also depends on the physiological state of the cell, meaning whether it is present in its vegetative state or any of the other adapted forms like spores. Yeast is generally more sensitive than bacteria. It is not effective against spores, so you can consider it as a pasteurization methodology. These days, it is used in food dehydration as it is able to reduce the quality changes associated with dry and semi-dry products. So when you can apply pulsed electric field to retain organoleptic properties and nutritional content of the food product, you will obviously opt for this methodology in food dehydration. A, because it is going to be very consumer friendly. B, it is going to reduce the energy costs associated with drying process. And finally, because it reduces the dehydration time or the drying time. It is used in products like raw and constituted apple juice, skimmed milk, beaten eggs, and peach juice. Thank you for staying with me through this video. In case you have any queries, please feel to put them in the comment section and I'll get back to you. In my next video, we'll be talking about yet another interesting preservation technology called as oscillating magnetic field. Until then, Merci beaucoup.